Couple of items. The learning path is up for your first exam. If you're having any problems with some of the things with Connect or the lab, please be sure to contact Liz first about that. If you're having any difficulty with Liz, you can, of course, contact me. I'm getting actually pretty good with the Connect stuff, too, so I can answer some of those questions. Because LearnSmart Labs is a little different than Phil's, and it is based on a system that will not allow you to proceed to the next level, so to speak, until you've exhibited complete mastery of that level. So a lot of you are seeing not 100% completion, even though you have completed the program. What it's telling you is that it doesn't think that you completely understand some of the nuances of that learning experience. And so it wants you to go back and rehearse or repeat those areas where you actually had some initial errors. Now, you don't have to do that. If you have 90% or better in your score on the LearnSmart Labs, that qualifies as completing it as far as we're concerned. So those of you that are getting fixated on that 100% for how many points? 1 point? I wish you would fixate so intensely on your exam as much as you are on that 1 point item, because that exam is worth 100 of those 1 points. So if you were that fixated on your exam, you would have time for nothing else in your lives, correct? So realize, also prioritize a little bit. If you find that, you know, this isn't getting me where I want, that's okay, because that's another thing, remember, we're trying to teach you in A&P is prioritize. So 100 points versus 1 point, and you should be spending your time on those things which give you the biggest bang for your buck. As you do know, the bulk of your grade comes from your lecture exams. So you always want to work hard on getting good lecture exams. Your labs can bring you down if you don't perform at least average in the labs. So make sure your performance in lab is at least average, and you're going to find you're going to do well, except there is one thing in lab which can definitely bring your grade down. That's that lab practical. So everything you do in lab, remember, should be geared towards prepping yourselves for the end of the semester experience, which is the lab practical. And that's one reason why we have you actually work on making lab composition notebooks for yourself, because that writing down is a good rehearsal to help you figure out how to study for that lab practical. The other thing too is I want to make sure you guys here in the face-to-face class know there will not be a formal lecture here in the lecture hall on Monday. So if you want to come here and study, that's more than acceptable for you, but there will be no formal lecture on Monday. The lecture will be actually pre-recorded for you and available to you over the weekend. And that's something that is designed to where if we don't finish up our endocrine before the weekend, I'll get you that lecture out by sometime Friday, and then you'll be able to have that lecture to help you study for the upcoming exam. So that's one of the reasons for no face-to-face class, other than if you want to come in for a study group, and I'll see if I have one of my pro section students that might want to come in and help facilitate your study group if you want to actually show up here on Monday. So keep that in mind. Now to the notes. So we're on the endocrine system, which is used to help create a homeostatic system for us. We have the nervous system, works together with the endocrine system. We get an immediate response with the nervous system typically, and then we get a delayed response with the endocrine system to help take care of what I call background issues for us. Now the hormones will activate their target cells via two different mechanisms, and it's based upon whether they're amino acid based or if they're steroid based. And we're going to talk now about this second messenger system. The second messenger is cyclic AMP. The first messenger is, of course, the hormone. So what happens is amino acids will get to their target cell, and they will bind to a membrane receptor. This receptor will then, it's actually very close to an internal protein called a G protein. And this G protein is activated by this membrane-bound receptor hormone complex. The G protein will then in turn activate adenylate cyclase. And adenylate cyclase will then convert ATP into cyclic AMP. Cyclic AMP is the second messenger in the system. Hormone's the first, and cyclic AMP, we're inside the cell right now, is going to activate certain types of protein kinases. So it's going to be very dependent upon what the cell is. And so when it activates these protein kinases inside the cell, this activity of the protein kinases will then activate or deactivate some other intracellular enzyme. This is something that we were talking about previously when we were talking about enzyme activity, activate or deactivate. So this is something that the second messenger system actually initiates. So cyclic AMP is the second messenger. It activates protein kinase, which will then activate or deactivate some other intracellular enzyme. This is referred as a cascading event. One thing causes this to happen, which will cause this next thing to happen, which will cause this next thing to happen, and so it's down like a waterfall. This is what is the destined event to actually do. So cascading event. This is a term that we'll also see when we deal with coagulation, because it is a cascading event. An activation of one will then activate the next thing, which will then activate the next thing. So those are amino acid-based hormones. Direct gene activation is then what we see with our steroid-based hormones. So this is the mechanism that they do or use. Steroids are fats or lipids, and what are cell membranes made out of predominantly? Lipids, phospholipid bilayer. And remember, there is actually cholesterol within the phospholipid bilayer. So we are taking steroid-based hormones, which are cholesterol-based hormones, and they're actually able, even despite their size, they're able to actually wiggle through that phospholipid bilayer because they're lipophilic. They like fats. Fats like fats. So they're able to actually move through that membrane relatively easily, despite their size. Once inside the cell, there's going to be this receptor that they are going to actually then bind to. So they wiggle through the membrane, get inside the cytoplasm of the cell, find a receptor that's destined for them. They bind to that receptor and create that hormone receptor complex. This hormone receptor complex is then transported into the nucleus. And once inside the cell nucleus, it will attach to the DNA in that cell and activate a gene. So that's the mechanism for our steroid hormones. How they actually will activate or impact their target cells. Now we're into control of hormone levels. And most of our hormone levels are controlled by a negative feedback process. And intuitively, you know this. When you have a meal, what happens to the glucose level in your bloodstream? It goes up. what then is going to happen in a normal individual? Insulin's being released. Insulin is a hormone, and it's released into the bloodstream. When insulin is released into the bloodstream, what happens to your blood glucose levels? Goes down. What happens to your insulin levels? Goes down. That's a perfect example of negative feedback. So it is released and gets to a certain level, does its job, and then we have to stop its release. We don't want to have that continuous release of insulin, because then we'd have our blood glucose levels crater on us, and then we'd actually go into an insulin coma, because the brain is an obligate feeder of glucose. And so then you're starving the brain, and it will literally pass out from lack of glucose for you. So negative feedback is the most common control mechanism for homeostasis for our body, as well as for hormone release and hormone levels. The synthesis, and therefore the release of our hormones, is going to be dependent upon what stimulates the endocrine gland, and we're going to talk about that in just a moment. Before we do that, let's talk about, once the hormone levels are up and circulating, how do we get those hormone levels down besides no longer releasing them? And so that's what elimination is all about. Your hormones can actually be enzymatically broken down in the liver. This is a very, very common reason why we need the liver, amongst many other things. The liver is a processing plant. And so it will take these hormones that it is actually designed to actually break down or degrade and break them down for us. Another thing that's really great and why we need kidneys is kidneys are also a wonderful organ, because you can put excess hormone levels into your urine and you can literally pee them out. So that's a very common mechanism. For example, Premarin. Does anybody know what Premarin is? Premarin is an estrogen women used to take because before they started the estrogen scare for breast cancer and stuff, women used to take Premarin to help them with the menopause symptoms. And the menopause symptoms can be very distressing to women. Hot flushes, unable to sleep at night, moodiness, irritability, other things actually associated with that. And so they were prescribed Premarin. Premarin is short for pregnant mare's urine. You following me? So in the urine is estrogen. And so pregnant mares have a high level of estrogen hormone, and they were actually taking their urine and processing the urine, taking everything out, and just where they actually had just the estrogen compounds. And this was prescribed in a pill. Yes?
Did the women know that's what they were taking?
They were taking hormones. And it made them feel better, because they wouldn't take them if it made them feel bad. So they were taking hormones that made them feel better. And this also had a beneficial effect on delaying osteoporosis and things like that. But you have to realize, you guys take a lot of things over the counter, and you should really do some research on what you're taking over the counter. One of the things about the supplement industry, it's not regulated. You know what that means? You have to trust them to be honest, that they are going to be more honest to you than to their bottom line. And so that's why there's actually a lot of lead in a lot of supplements over the counter. And arsenic, lovely heavy metal poisonings are in all kinds of over-the-counter supplements. So you actually really have to do your research if you're taking some of those things. So we've been ingesting all kinds of weird things as a society for a long time. So Premarin wasn't considered all that abnormal, because also, we'll get down to diabetes in a little bit, we used to inject people with insulin from pigs and cows. And now we no longer do that, because we have more modern mechanisms in which to actually make that insulin. So you have to realize we have systems inside us that help to protect us from ourselves, so to speak. So the kidneys are involved in excretion of various hormones, which is one way to get them out of your body. And also, of course, in the case of steroid hormones, target cell uptake. So that takes that steroid, takes it inside the cell, binds that receptor complex that will then move it into the nucleus, and then ultimately that hormone will be degraded down by the cell itself. So that's how hormones are typically eliminated. Now, the mechanism to turn them on we just discussed. So let's take a look at the cellular level. We discussed that with cyclic AMP and then the direct gene activation. But let's talk about what this stimuli is going to require in order to get this process initiated. So endocrine gland stimuli, what gets these glands initiating the process of making their hormones for their target cells? Hormonal. Another hormone controls a gland's release of a hormone. And this is important. This is defined as a tropic hormone. A tropic hormone is a hormone that will cause an endocrine gland to release its hormone. And you say, well, that's kind of weird. No, it's not, because actually that's a lot of our anterior pituitary hormones are actually released. So there's tropic hormones that are acting on the anterior pituitary to cause it to release its hormones. And even those hormones can be tropic because they're going to cause another downstream endocrine gland to produce hormones. You'll see RH for releasing hormone and IH for inhibiting hormone. So these are some definite tropic hormones that are going to actually be released by the hypothalamus onto the anterior pituitary, and we're actually going to see that in just a moment. Humoral. So a hormone can make an endocrine gland release its hormone. Humoral. The level of something in your body fluids will control the gland's release of that hormone. And we just discussed what controlled the release of insulin. Blood glucose levels. So blood glucose is an example of something that is humoral. It's in the fluids or humors of our blood, and it will cause the release of insulin when it's up. Another example of a humoral control of a hormone's release is potassium levels. When potassium levels are up, it will cause the adrenal cortex to release aldosterone. This is the actual major control of the release of aldosterone. Elevated blood potassium levels. Well, if aldosterone is released for elevated blood potassium levels, what do you think aldosterone impacts? Blood potassium levels. Just like insulin affected blood glucose levels, aldosterone is going to impact blood potassium levels. And if elevated potassium causes aldosterone release, what will stop aldosterone release? Normal potassium levels. And so aldosterone will cause the kidneys to put potassium in the urine, and when it does so, because it's an ionic exchange process, we will retain sodium. So this is a very important mechanism for homeostasis, and we'll talk about that in just a moment. The final mechanism that will impact endocrine glands, causing them to release their hormone, is neural. So the nervous system can cause hormone release, and this is indeed what we see with the adrenal medulla. When we impact the adrenal medulla, when we say that we want you to release your hormones, the hormones in the adrenal medulla are epinephrine and norepinephrine. And it will cause those neurotransmitters. Isn't that what you guys learned previously? These are neurotransmitters. Well, they're more than just neurotransmitters. They're also considered hormones because a gland is putting them into the bloodstream. When we put epinephrine and norepinephrine into the bloodstream, it will circulate on around, and it will hit all kinds of target cells, helping to prolong the effect of what branch of the autonomic nervous system? Sympathetic, fight, flight. So this is an important mechanism for control. So we see in our little pictures here, here's a hormonal situation. Thyroid stimulating hormone is another tropic hormone, which will impact the thyroid gland and cause that gland to release thyroid hormone into the bloodstream. We just talked about blood glucose for our humoral, and we just mentioned the autonomic nervous system, the sympathetic branch of the nervous system, which is also part of the hypothalamus. impacting the release of epinephrine and norepinephrine from the adrenal medulla into the bloodstream. Let's start looking at some of the specific organs of the endocrine system. The pituitary is also called the hypophysis, and it is attached by a stalk that is also called the infundibulum. You might as well learn some of these names because you're going to encounter them out in your career somewhere. The infundibulum is attached to the inferior side of the brain, more specifically the hypothalamus, which is what our little picture here shows. So hypothalamus, little stalk called the infundibulum. And so the pituitary or hypophysis has two distinct areas, and we call them lobes. We have an anterior pituitary and a posterior pituitary. The anterior pituitary is also called the adenohypophysis. And the posterior pituitary is called the neurohypophysis. So the anterior pituitary looks very different than the posterior pituitary, and it's easily seen on a lower power view. The anterior pituitary will typically be darkly staining because these adeno means glandular, by the way. Adenocarcinoma is a glandular cancer. So adenohypophysis, these cells are glandular cells, and they're nice and round. The classical name for them is cuboidal cells because they're just they're cuboidal cells without any corners, in other words. But they're nice round cells, and we're going to take a look at them in just a moment, and they have certain staining characteristics based upon the type of hormone they're actually producing. The neurohypophysis will be paler in appearance and will be full of glial cells that are responsible for taking care of the axonal endings that are actually located in the neurohypophysis and contain hormones. The hypothalamus is responsible for all of these releasing hormones and inhibiting hormones. So we have TRH for thyrotropin releasing hormone, which is going to impact the release of thyroid stimulating hormone from the anterior pituitary. We have PRH, prolactin releasing hormone, cause the anterior pituitary to release prolactin. Then we have GnRH, gonadotropin releasing hormone, for release of luteinizing hormone and follicle stimulating hormone. We have CRH, corticotropin releasing hormone. Even in its name, it tells you it's a tropic hormone because it has tropin in its name. And that is going to cause the anterior pituitary to release adrenocorticotropic hormone, ACTH. We have growth hormone releasing hormone, which is going to cause the anterior pituitary to release growth hormone. And then we have some inhibiting hormones, just two, prolactin inhibiting hormone and growth hormone inhibiting hormone. So we have a nice cartoon here that shows you the anterior pituitary is going to be impacted by those hormones, and TSH is going to be released from the anterior pituitary. Prolactin is going to be released from the anterior pituitary. FSH and LH is released from the anterior pituitary. ACTH is released from the anterior pituitary, and growth hormone is released from the anterior pituitary. So those are all of the hormones that we will find being released from that anterior pituitary, and we're going to take a look at that in a little bit more detail now. Our notes here say two main lobes, anterior pituitary, also called adenohypophysis, posterior pituitary, called neurohypophysis. With our anterior lobe, remember, adeno is glandular. We're going to have three main types of cells that we want you to know. We have acidophils, we have basophils, and we have chromophobes. So they're named for the stain that impacts the visibility of the cell. So acidophils will take up an acid stain, basophils will take up a basic stain, and chromophobes will not like stain. Chromo is for color, and so it doesn't like to stain. And so this is a very nice histology view here. I know it's not part of your notes, but it should be part of your histology atlas in your lab composition notebook. So this is something that's relatively familiar with you if you've been preparing for your endocrine lab at all. So this is designed to show you acidophils, basophils, and chromophobes. And right smack dab in the middle, almost in the middle to me, here's a beautiful example of a chromophobe. Excellent. And then I would say this cell here is a lovely example of a acidophil. That's right, red or pink stain. And then some nice basophils would be something like that. Now, it isn't that there aren't other good examples on this slide. It's just, to me, these are the ones that look kind of the best. And so that's what you want to do whenever you make a histology drawing in your lab composition notebook histology section. Make sure you have a couple of really good examples, and then of course add a couple of examples that are not so good, so just in case you know what your variability of the terrain is going to be like. The acidophils are responsible for releasing these hormones that are related to this acronym, and the basophils are responsible for releasing these hormones related to this acronym. A here is for acidophil, and B here is for basophil. So G stands for growth hormone, P stands for prolactin. Those are the hormones released from your acidophil cells. In your B-flat acronym, F stands for follicle stimulating hormone, which is a gonadotropin type of hormone. L is for luteinizing hormone. A is for ACTH. It's easier to say that than adrenocorticotropic hormone. And T is for TSH, or thyroid stimulating hormone. Chromophobes, we don't know what they are there for yet. We haven't quite figured them out. So those are the three main cell types we want you to be able to identify in the drawings that you have in your lab composition notebooks for the anterior pituitary. So growth hormone, what is it good for? Well, it's good for general body growth. Thyroid stimulating hormone is abbreviated TSH, and that stimulates the thyroid gland to release thyroid hormone. H is our standard abbreviation for hormone. So TH is just shorthand for thyroid hormone. Adrenocorticotropic hormone, or ACTH, controls the release of hormones from the adrenal cortex. Gonadotropins, our two gonadotropins are FSH for follicle stimulating hormone and LH for luteinizing hormone. Macula lutea, so it's like luteinizing hormone. And these are responsible for controlling the release of hormones from our gonads. So testosterone for men and estrogen and progesterone for women. Prolactin is going to stimulate milk production after childbirth. So those are the hormones from the anterior pituitary. Now, the posterior pituitary, also known as the neurohypophysis, is going to contain the cell bodies that are responsible for these hormones actually live in the hypothalamus. So cell bodies axons pass through infundibulum, where our axon terminals or endings, which are storing hormone for eventual release, because it's going to be the hypothalamus that's going to actually determine that the hormones are going to be released from the posterior pituitary. Throughout the posterior lobe, the main cell is pituocytes. The pituocytes are glial-like cells. Do you remember what glial is a generic term for? It's kind of like support cells for neurons. So these are, the posterior lobe is full of these pituocytes, which are basically support cells for the axonal terminals, which are actually storing the hormone until it's time for them to actually be released. So that's what the majority of the posterior lobe is. You will not be able to easily make out axonal terminals storing hormones. So these axonal terminals are going to store hormones called ADH, which is short for antidiuretic hormone. This is going to impact the kidneys. Are the kidneys going to make more or less urine when antidiuretic hormone stimulates them? Less. Diuresis is to pee or urinate, and antidiuresis means don't pee or urinate, which means make less. Why would we want to have a hormone like this telling our kidneys not to make so much pee? Our own body's fluid balance. So if you are dehydrated, what do you think the antidiuretic hormone level is? High. Absolutely, because it wants you to actually not make urine because it needs to keep it inside your body for this dehydration status. Now, oxytocin is the other hormone that's released from the posterior pituitary, and it will stimulate contraction of smooth muscle in a woman's uterus or her breast. And this is a rare example of positive feedback control. So this is the one that's kind of the outlier, but this is designed to help facilitate delivery of children, and then after the child is born, then it's designed to continue actually to eject milk from the breast so that the baby doesn't have to work so hard while actually breastfeeding. So it actually contracts the smooth muscle around the lactiferous units of the breast, and literally women have actually almost seen breasts squirt from their nipples without any impact sometimes when babies cry. And that's why they have those nipple shields for that very situation. This is a flow chart. So this is a flow chart of things that we were just looking at above, of the cartoon picture that we were looking at. Hypothalamus is the ANS control center, isn't it? And so it's ANS control center, and it's a big controller of the endocrine system, not just of autonomic nervous system functions. So corticotropin releasing hormone, thyrotropin releasing hormone, gonadotropin releasing hormone, growth hormone releasing or inhibiting hormone, and prolactin releasing or inhibiting hormone. These are all tropic hormones. Tropic hormones cause the release of a hormone from an endocrine gland. Your pituitary gland is an endocrine gland. The anterior aspect of the pituitary gland is where all those hormones impact and will cause the downstream release of another hormone, adrenocorticotropic hormone, thyroid stimulating hormone, luteinizing hormone, and FSH, or follicle stimulating hormone, growth hormone, and prolactin. Are all of those tropic hormones? No, but some of them are, yes? The first three are tropic hormones. ACTH acts on the adrenal cortex to cause it to release hormones which have a broad category of corticoids. That's what ACTH is best at causing the release from the adrenal cortex. Thyroid stimulating hormone will cause the thyroid gland, which is an endocrine gland, to release thyroid hormone, which will affect general body metabolism. Luteinizing hormone and follicle stimulating hormone act on the gonads, testes, or ovary to cause them to release their sex hormones. So ACTH, TSH, LH, and FSH are all tropic hormones. However, growth hormone and prolactin are not tropic hormones because they do act on body tissues, but they don't cause these body tissues, for example, with growth hormone, to release another hormone. They are promoting tissue growth for growth hormone and for prolactin, milk production after birth of the child. So that's how the anterior pituitary's flow chart is. Now let's take a look at the posterior pituitary or neurohypophysis and realize this is lovely little axons, isn't it? Coming from the hypothalamus in the infundibulum down to the posterior lobe, causing the neurohypophysis to release ADH and OT, oxytocin. Are those tropic hormones at all? No, these are not examples of tropic hormones. So water reabsorption, which is not making much urine, and contraction of the uterus and let down for milk. This is an ANS sympathetic direct connection. This is truly, the hypothalamus has essentially a sympathetic direct connection to our adrenal medulla because the adrenal medulla is neuronal tissue. It's neural tissue. And when the hypothalamus says fight, flight time, it will cause the adrenal medulla to release epinephrine and norepinephrine into the bloodstream. So that flow chart is something to spend a little bit of time on to see if you can actually create it yourself. Let's talk a little bit now about some other glands of the endocrine system, the thyroid gland, parathyroid, and then the adrenal glands. We'll almost be done for today. Thyroid gland. Let's take a look at this histology picture over here. How would you describe that? Let's talk about, first off, let's talk about cellularity. Compare the thyroid gland to the cellularity of the parathyroid glands. Which one is more cellular? Parathyroid. Big difference when you take a look at one versus the other. So this is how you actually approach when taking a look at your histology. Why do you know the top one is thyroid? Big pink things, right? Big pink things surrounded by essentially a single layer of what type of cells? Cuboidal cells, and they are very flattened cuboidal cells, correct? Because they aren't squamous cells because you can see cytoplasm. Are you supposed to be able to see a bunch of cytoplasm with squamous cells? No, so you just see a nucleus and it's kind of squished itself. So these are cuboidal cells because we can still see some cytoplasm. The reason they're flattened is because there used to not be a bunch of pink stuff in the middle. And then they started putting this pink stuff in the middle, which is called colloid. And as they put more and more pink stuff in the middle, they got pushed back. So that colloid is making them flat, is what it is. And so if you were to describe the essential structure of this structure is, the basic structure is colloid-filled follicles. Follicle cells, which are the cells around the perimeter of that colloid, are the ones that produce thyroid hormone. And they produce it in its inactive form initially. And store it in the colloid. So the stuff in this colloid material is inactive. It's not fully activated thyroid hormone. It's kind of like a precursor to thyroid hormone. And We know thyroid hormone is our major metabolic hormone. And is released in the bloodstream by elevated levels of TSH. If TSH levels are low, what does that mean about thyroid hormone release? It's not high, like when TSH levels are high, correct? So thyroid stimulating hormone, when it's elevated, it will tell the thyroid gland, release more thyroid hormone. When thyroid hormone levels are adequate, TSH levels go down. That's our negative feedback mechanism. The important thing about thyroid stimulating hormone is it happens to be a fat-soluble hormone. So it needs carrier proteins to actually be transported in the bloodstream. The protein happens to have a really interesting name. It's called TBG. It's called thyroid binding globulin. So when the thyroid gland releases active thyroid hormone, so thyroid hormone actually is T3 and T4 whenever it's released. The 3 and the 4 are related to the number of iodine complexed with that hormone. So it needs iodine to be active or activated. So T3 means how many iodines? And T4 means how many iodines? And T4 is often called thyroxine. And this is a pill or supplement that are given to women and men who happen to have low thyroid function or hypothyroidism. So it's easily supplemented in an oral form, and the pill is called thyroxine, and it's just T4. The thyroid hormone is T3 and T4. What's fascinating is the bulk of the hormone that's released by the thyroid gland is mostly T4. The active hormone is T3, but the cells are actually programmed to convert T4 to T3 when it gets to them. So we can give people T4 because the bulk of our thyroid hormone is T4, and at the cellular level, the cells will convert T4 to T3. It's easy to knock off an iodine than to figure out how to add one to it. So T3 is the active form, and our cells actually make T3 out of T4. Yes?
I was just going to add that the Phils said that T4 is also has a longer life.
The half-life, it does have the longer half-life. The other thing too is it's only the free hormone that the cells can use. So our binding globulins, anything that actually is binding our steroid-based hormones, are there to help to prevent hormone breakdown or degradation. And so these hormones are present in the blood as bound or free. The free is very small, the bound is very big, and it's the free that the cells will actually use and actually be transported into the cell. So this is a very important mechanism for preservation of our fat-based and steroid-based. This one is not a steroid-based, it's a fat-based hormone. So T4 is thyroxine. And this is what people take. euthyroid or hypothyroid. There's some other cells in here besides follicle cells, correct? They're called parafollicular cells. And the parafollicular cells are in this general area. They're in between the follicles. So parafollicular cells are not follicle cells. They're in between the follicles. So make sure in your drawing that you have some parafollicular cells so you can identify those. The parafollicular cells, let me go ahead and make some room here. The parafollicular cells produce a hormone called calcitonin. What do you suppose calcitonin has some sort of impact on? Calcium. Yes, the name actually tells you something about it. And so what's nice about calcitonin, it will actually inhibit osteoclast activity and stimulate osteoblast activity. So that's what it does, inhibits osteoclasts plus stimulates osteoblasts. What will that do to a calcium level in the bloodstream? If you inhibit clasts and stimulate blasts, which ones are responsible, first off, which ones are responsible for breaking down bone? Clasts. If you break down bone, that means you're releasing calcium into the bloodstream, right? And if you're a blast, that means you're building bone and you're putting calcium into bone. So overall, what does this do to a calcium level? Lowers it. So calcitonin is released when calcium levels are high, and it will lower it. So inhibits blast and clasts. So released by high calcium levels to decrease blood calcium levels. This is more important in kids than it is in adults. This mechanism doesn't seem to work too well for adults. The mechanism that works well for calcium homeostasis in adults is due to the parathyroid glands. We'll talk about that next time. And stop for today and do a clicker.